Usually when we think about a walk through a quiet forest, there's this underlying expectation of stillness.
Oh, absolutely.
Yeah, you step off the pavement onto the dirt, and you feel like you're entering a sanctuary, right?
Mm.
Where everything's operating on this glacial, very passive time scale.
Right, yeah. Because our metabolic rate is just so different, we assume a plant is just, sitting there,
Yeah,
just enduring whatever happens to it.
exactly. You look at a birch tree with that stark, brilliant white bark peeling off in paper-thin strips in the wind, and you just see a landscape painting, a static object holding its ground. It's calming because it feels totally inactive.
But the closer you look, the more that illusion just completely shatters.
It really does. When you actually step into the world of molecular biology and ecological physiology and subterranean mycology, that quiet forest completely disappears.
It vanishes.
What you're actually walking through, what you're stepping directly on top of, is a bustling, highly intelligent, chemical, and communicative superhighway.
The sheer volume of data processing happening under your boots is staggering. I mean, today, we're going to use the birch tree, the Betula genus, as our lens to examine exactly how complex that processing gets.
Welcome to the deep dive. We are taking you on a massive, multidisciplinary exploration today. We're going to completely deconstruct the birch tree.
We are moving far, far beyond its status as just a pretty ornamental tree in a suburban front yard.
So true. We're going to start at the microscopic level, looking at the exact DNA methylation patterns that govern its phenotypic expression. We'll examine the allometric physics of its wood.
Right, the actual mathematical algorithms it uses to survive sub-zero temperatures.
Yeah. And then we're going to break down its chemical medicinal armor, trace the amino acids flowing through its subterranean neural networks, and finally look at how early humans intuitively recognized all of this biology and elevated the birch to the mythological status of the world tree. Okay, let's unpack this.
Well, to truly understand the birch, we have to start at the absolute base code, right? It's molecular genetics.
The blueprint.
Exactly. And rather than looking at a standard green birch, it's actually much more illuminating to look at a highly specific mutation. There's this fascinating variety called Betula pendula purple rain.
Great name, by the way.
I know, right? And as you might guess, it has these incredibly dark, striking purple leaves. But during a recent tissue culture experiment, a line of this purple birch, which was designated the NZ line, underwent a spontaneous phenotypic shift.
Spontaneous meaning what? It just happened on its own.
Exactly. It produced a mutant line called NL, which completely lost its purple coloration. I mean, it reverted entirely to green leaves.
Now, when you see a massive morphological shift like that, a plant entirely losing a dominant pigment, the immediate assumption is that the DNA itself broke, right?
Yeah, you'd assume a mutation in the sequence, a
Right.
deletion or something.
Right. But when the genome of this green NL mutant was completely resequenced, the base code was pristine.
Perfectly intact.
There were 0 single nucleotide polymorphisms, no insertion or deletion mutations in the pigment pathways. The genetic architecture to produce purple leaves was completely there. So the shift wasn't genetic at all, right? It was entirely epigenetic.
Right, and we're looking specifically at the suppression of flavonoids here.
Break that down for us. What are flavonoids?
So flavonoids are this massive class of compounds in plants defined by a C3C6 structure.
Which means?
It means you have 2 benzene rings, the C6 parts, linked together by a 3-carbon chain. It's a very specific chemical backbone. And the specific subcategory of flavonoids responsible for that deep purple hue are the anthocyanins.
Got it. So the purple rain birch makes a lot of these anthocyanins.
Oh, it pumps them out in massive quantities. But the NL mutant just stopped. And the mechanism for that shutdown is just a masterclass in epigenetic regulation. It all centers around a regulatory system called the MBW complex.
Which is a triad of transcription factors, right?
Yeah.
I've got it here, MYB, BHLH, and WDR proteins.
Exactly. These 3 proteins, they have to bind together to form a functional complex. Once they form, this complex essentially walks down the DNA strand, finds the specific promoter regions for the anthocyanin biosynthesis genes, and switches them on.
Like turning a key.
Right. But in the green mutant, the transcription factor that failed was the MYB component, which is coded by a gene called BPMYB113.
But it didn't fail because the gene was broken, Right, we already established that DNA was fun.
Exactly. It failed because of hypermethylation in the CHH context.
Okay, let's get into the weeds on that CHH context because that's where the actual physical mechanism lives.
Yeah, this is so cool. So in mammalian epigenetics, like in humans, we're usually talking about methylation at CG sites. That's a cytosine followed by a guanine.
Right, pretty standard.
But plants have this highly complex, specialized system where they can methylate cytosines in a CHH sequence.
Wait, CHH, meaning what exactly?
Meaning a cytosine followed by two nucleotides that are anything except guanine. So like adenine, thymine, or another cytosine.
Oh, wow, so they have way more targets.
Way more. And this is primarily driven by a process called the RNA-directed DNA methylation pathway, or RDDM.
RDDM, okay.
Basically, small interfering RNAs act like homing beacons. They guide these methyltransferase enzymes exactly to this BPMYB113 gene, and they just slap methyl groups all over its promoter region.
And physically, what does that methyl group actually do? I mean, it's just a carbon atom bonded to 3 hydrogens. How does something that tiny shut down an entire leaf's color?
It's all about physical space. It changes the physical shape of the DNA. It alters the chromatin structure, packing it so tightly that the cellular machinery, the RNA polymerases, literally cannot access the gene to transcribe it.
But it blocks the reading head.
Exactly. It's like taking a page of an instruction manual and gluing it shut. You have the instructions, but you can't read them. And because that one MYB transcription factor couldn't be produced, the entire downstream assembly line collapsed.
Right, the entire phenolin metabolic pathway.
Yes, which requires a highly orchestrated sequence of enzymes like PL, C4H, 4CL, CHS, CHI. All of those were drastically downregulated.
So without that master switch, the tree just couldn't produce chalcone, which is the precursor to the anthocyanins.
Precisely.
It reminds me of player piano. You know, the green mutant has the exact same piano keys, the exact same DNA sequence as the purple birch. The hardware is totally identical.
I love that analogy.
But the sheet music being fed into the piano has been punched with different holes. The RDDM pathway went in and essentially covered up specific holes on the roll. So the piano physically cannot play the purple chord anymore. It defaults to the green chord.
That is exactly what's happening.
But what I find completely mind-bending about this is the trigger. Like, this wasn't an evolutionary adaptation over thousands of years. This epigenetic shift happened because of the trauma of human-induced tissue culture.
Yes. And this is a huge realization in botany right now. The stress of being excised from the parent plant, placed in an artificial agar medium, and bombarded with exogenous growth hormones, it induced a massive stress response.
The plant basically panicked.
Yeah, its epigenetic machinery panicked and started rapidly remodeling its chromatin just to survive the sheer shock of it all. And in doing so, it accidentally silenced the pigment pathway.
Wow.
But what's fascinating here is that this suppression is highly dynamic.
So it's not a permanent weld.
Not at all. Young leaves emerging on this green mutant actually still show a pale purple hue.
Oh, really?
Yeah, as the leaf expands and matures, the RNA-directed methylation pathway basically has to catch up. As it does, the chromatin tightens and the color fades completely to green over time.
That is wild. We are watching the tree chemically rewrite its own operating system in real time as the leaf develops.
Exactly. It proves that epigenetic silencing isn't just a static one-time event. It's a fluid, continuous metabolic effort by the plant to manage its environmental stress.
Now, that dynamic regulation is incredible when you're looking at the delicate tissue of a leaf. But the exact same level of hyperspecific genetic control dictates the physical load-bearing architecture of the wood itself.
Oh, absolutely.
Because the trunk of the birch isn't just dead scaffolding holding leaves up to the sun. It's a highly organized, rapidly dividing structure.
Right. And to understand the trunk, we have to look at xylogenesis.
Which is the biological formation of wood.
Correct. Right beneath the bark of the birch is a microscopic layer of actively dividing cells called the cambium, and how those cambial cells divide dictates the entire physical reality of the tree.
And just like the leaf color, this is tightly controlled by transcription factors and hormones.
Yes. Let's look at a highly prized anomaly in the woodworking world. It's called the Carillion birch.
Oh, yeah. If you've ever seen a high-end custom knife handle or like the dashboard of a bespoke luxury car, you might have seen this.
It is stunning.
It doesn't look like normal wood. It has this incredibly complex, rippled, chaotic, swirling grain pattern. It's often called figured wood.
Right. And for centuries, people had no idea why a random birch tree would grow this wildly beautiful twisted wood while the tree right next to it grew perfectly straight.
But molecular genetics has finally cracked that mystery, hasn't it?
It has. And it comes down to a family of NAS domain transcription factors, specifically the VND, NST, and SND genes.
Okay, what do those do?
These are the master regulators for secondary cell wall biosynthesis. They literally control how the tree lays down cellulose, hemicellulose, and lignin.
The building blocks of the woods.
Exactly. Now, in a normal straight-grained birch, a gene called BPVND7 is operating at peak efficiency. It enforces strict discipline in the cambium.
Discipline meaning order.
Yeah. It forces the cells to undergo what we call paraclinal divisions, meaning the cells divide perfectly parallel to the surface of the trunk, which creates those neat, concentric, straight rings of wood we are all familiar with.
But in the Karelian birch, that discipline completely collapses.
Completely. The cambium just goes rogue. The cells start dividing anticlinally or transversely, meaning perpendicular to the surface.
Oh, so they're turning sideways.
Exactly. They fold in on themselves. They create pockets of bark trapped inside the wood, creating that swirling, starry pattern. And the root cause of this structural chaos is a localized drought of a very specific hormone.
Auxin.
Yes. Indole-3-acetic acid. It is arguably the most important hormone in plant structural development.
It's like the master architect.
Yeah, it dictates vascular patterning, telling the cells exactly what orientation they need to take. In the Karelian birch, there is a severe localized downregulation of an auxin-dependent transcription factor called BPARF5.
BPARF5. And its direct target gene, BPHB8.
So the signal just drops out.
Precisely. Because those auxin signaling genes are suppressed, the spatial awareness of the cambium breaks down. The cells essentially lose their compass.
Right.
They don't know which way is out and which way is up, so they just divide chaotically.
That is amazing. So that beautiful, highly expensive figured wood is, biologically speaking, a severe hormonal glitch. It's a failure of spatial signaling.
That's exactly what it is.
But altering physical structure isn't always a glitch, right?
Mm.
In some of the harshest environments on Earth, the birch intentionally manipulates its own physical architecture, its stature, as a highly calculated thermodynamic survival mechanism. Yes, and this is where we move from molecular biology into ecological physiology. If we look at studies from the alpine tree line ecotones, specifically research done on Changbai Mountain in northeastern China, we see a masterclass in what's called stature plasticity.
So they're tracking these trees going up a mountain.
Right. They tracked birch trees growing along a brutal, steep altitudinal gradient, starting at 1,700 meters above sea level and going right up to the absolute physiological limit of tree growth at 2,050 meters.
And up there, I mean, the air temperature is lethal, the wind shear is intense, and the growing season is incredibly short.
It's a brutal environment.
To survive that, the birch calculates a mathematical scaling relationship between its height and its diameter, right? It's allometric growth.
Yes, and researchers found a very specific, hard mathematical breakpoint at exactly 2.25 meters in height.
2.25 meters. So what happens at that height?
Well, below that breakpoint, the birch behaves entirely differently than it does above it. Birch saplings that are under 1.4 meters tall adopt a stunted morphology.
Stunted like a bush.
Exactly. Instead of trying to grow a tall central leader branch, they invest almost all of their carbon reserves into radial growth, getting thicker, expanding their diameter, and throwing out multiple low stems.
And the physics behind that choice are actually brilliant. It's all about boundary layers and microclimates.
Tell them about the boundary layer.
Well, in the alpine tundra, the ambient air a few meters off the ground is brutally cold. It pulls heat away from the plant through convective cooling. But right at the surface, the ground absorbs solar radiation during the day. And then in the winter, the deep snowpack acts as this phenomenal thermal insulator.
Right. And the most vulnerable parts of a tree are its apical meristems, the actively dividing growing tips at the very ends of the branches.
Because if those freeze and die, the tree's upward growth is basically terminated.
Precisely. So by maintaining a stunted shrub-like form, the birch sapling physically decouples its vital organs from the freezing ambient air. It keeps its apical meristems tucked safely inside that warm microclimate near the soil and under the snowpack.
It's basically huddling.
That's a perfect word for it. It's an active metabolic decision to prioritize thermal safety over light competition.
But here is the genius of that allometric breakpoint, right?
Right.
Once a tree manages to successfully breach that 2.25-meter mark, once it is tall enough that it is permanently thrust out into the freezing mesoclimate, its strategy completely flips.
It has to. It can no longer hide in the boundary layer, so the tree becomes tapered.
It stops pouring carbon into diameter and starts aggressively pouring it into vertical height.
Yeah, it starts racing upward to outcompete neighboring plants for photons.
Which, raises an incredibly urgent question regarding climate dynamics.
Oh, absolutely.
If the birch is running this ancient, highly calibrated mathematical algorithm, chemically choosing between growing out to stay warm or growing up to catch light, how does rapid anthropogenic climate change scramble that math?
That is the cutting edge of tree line ecology right now. As mean annual temperatures rise, the thermodynamics of that boundary layer shift entirely.
Because the snow melts sooner.
The snowpack melts earlier, the ambient air warms. The breakpoint of 2.25 meters will likely shift higher, or the microclimate advantage will just disappear altogether.
Wow.
But the sheer competitive advantage of the birch's plasticity is what allows it to dominate up there.
Wow.
Consider the larch tree, which grows in the exact same high-altitude environments.
What does the larch do?
Larch trees respond to extreme cold by forming what we call a cushion shape. They grow incredibly dense and super tight to the ground.
Which sounds safe.
It is safe, but that cushion form severely limits their photosynthetic capacity and their growth rate. The birch, because of its multi-stemmed plasticity, can maintain a much larger leaf area index while still staying relatively low.
Ah, so it gets more sun while staying warm.
Exactly, allowing it to grow faster than the larch and dominate as the primary pioneer species.
So it basically outmaths the competition.
It really does.
But structural adaptation and thermal huddling are really only part of the survival equation. You also need active defense.
Oh, heavily active defense.
Because when you walk up to a paper birch and you run your hand along that peeling white bark, you aren't just touching dead cork.
No, not at all.
You are touching a staggering, hyperdense chemical arsenal.
We're transitioning now from structural physics into high-level pharmacology. The bark of the birch is heavily concentrated with pentacyclic triterpenoids.
Pentacyclic triterpenoids. Okay, what are those?
These are incredibly complex secondary metabolites. We're
Right.
talking about a massive 30-carbon skeleton that folds itself into 5 distinct interlocking rings.
Right.
Cyclic, right? 5 rings.
Right.
And in the birch, these compounds are built specifically on what's called a lupane skeleton.
A lupane skeleton.
Yeah, it's a specific 5-membered ring structure that features an isopropylene group attached to it.
Okay, to really ground this in reality for everyone listening, think about that powdery white residue that rubs off on your jacket when you lean against a birch trunk.
Yeah, that classic white powder.
That powder isn't dust. Up to 34% of the dry mass of that outer white bark is a pure, crystallized pentacyclic triterpenoid called betulin.
It's wild. The tree is literally sweating out crystallized armor.
Crystallized armor.
And the biochemical pathway required to synthesize betulin is deeply complex. It all runs through the mevalonate pathway, or MVA.
Walk us through that MVA pathway. How does it build this armor?
Well, the tree begins with a long hydrocarbon chain called squalene. Using an incredibly precise enzyme called lupeol synthase, the tree basically folds that squalene chain, snapping carbon bonds into place to form those 5 interlocking rings, creating a molecule called lupeol.
But lupeol isn't the final product, right?
No, it has to be oxidized.
Yeah.
And this is where a massive ancient family of enzymes called cytochrome P450 monoxygenases comes into play.
That sounds intense. Cytochrome P450.
They are intense. These enzymes use an iron-containing heme group to activate molecular oxygen and forcefully insert it into the lupeol molecule. Specifically, an enzyme called lupeol 28 monoxygenase oxidizes lupeol to create betulin.
Okay.
And if it oxidizes it even further, it creates beculinic acid, or BA.
And when you dive into the medical and pharmacological literature on betulin and betulinic acid, the data is just staggering.
It really is.
The tree is producing these compounds to protect itself from fungal rot and boring insects, obviously. But the mechanism of action translates profoundly to human medicine.
Beautifully.
Betulinic acid has been shown in multiple in vitro and in vivo studies to be a highly potent cytotoxic agent against human cancer cells, specifically melanoma, neuroblastoma, and leukemia.
And the incredible part is we know exactly how it works. Betulinic acid doesn't just poison the cell blindly, like traditional chemotherapy might. It induces apoptosis.
programmed cell death.
Right. It specifically targets the mitochondrial membrane of the cancer cell, altering its permeability.
So it punches a hole in the power plant.
Exactly. This causes the mitochondria to leak cytochrome C out into the cytosol, and that leak activates a cascade of caspase enzymes that systematically dismantle the cancer cell from the inside out.
While largely leaving healthy cells alone.
Yes, it's a highly targeted strike.
And on top of that, these lupane-type triterpenoids exhibit severe antiviral properties. Research shows they can inhibit the maturation of the HIV virus by blocking specific viral cleavage events, and they act as powerful, broad-spectrum antibacterial agents. I mean, the tree is literally wrapped in pharmaceutical-grade armor.
It is. There is, however, a significant biochemical hurdle when it comes to human pharmacology.
Which is what?
The physical structure of the lupane skeleton makes betulin highly hydrophobic. It heavily repels water.
Ah, so it doesn't mix with our blood.
Exactly. Because it doesn't dissolve in aqueous solutions, it has terrible bioavailability in the human bloodstream. If you just ingest raw betulin, your body can't absorb it effectively.
But pharmacologists are actively engineering workarounds for that, right?
Yes, through semisynthesis. Chemists basically take the natural betulin extracted from the birch bark and alter the functional groups on the rings.
Tweaking the chemistry.
Right. They create derivatives like betulonic acid, or they attach methyloxime groups to increase its solubility in water, all while maintaining that potent cytotoxic mechanism.
That's brilliant.
But even without modification, the raw extract is actually already in clinical use. It's the primary active phytochemical in episolvin, which is a topical gel approved in the European Union.
Oh, I've read about this. It dramatically accelerates the healing of severe skin wounds.
Exactly. It promotes keratinocyte migration, basically telling skin cells to move and close the wound faster.
So the birch is a chemical fortress.
Mm-hmm.
But here's where the biology takes a truly bizarre turn.
Oh, yes. The parasites.
Right. There are specific parasites that attack the birch, most notably the chaga mushroom, Inonotus obliquus.
Such a fascinating organism.
Chaga is a parasitic fungus that infects the heartwood of the tree. It rubs through the bark as this black and charred-looking mass and eventually kills the host.
Right.
But if you analyze the chemical composition of the chaga mushroom itself, it is packed with the exact same betulin and betulinic acid as the tree. How does a fungal parasite manage to synthesize the plant's own highly complex 30-carbon immune system?
It is one of the most breathtaking examples of convergent evolution and, honestly, biochemical theft in the entire natural world.
Because fungi and plants are not closely related at all.
No. You have a plant and a fungus. They belong to completely separate biological kingdoms. Their evolutionary lineages diverged hundreds of millions of years ago, yet the chaga mushroom possesses the exact genomic machinery, the exact mevalonate pathway enzymes we just talked about to synthesize pentacyclic triterpenoids.
So it's not just passively absorbing the betulin from the tree, like a sponge.
Well, it does absorb precursors from the wood, but it also manufactures them independently. As the fungus slowly digests the birch over years, it continuously encounters the tree's chemical defenses. Over evolutionary time, the fungus didn't just develop a tolerance to betulin, it actually integrated the chemical into its own biology.
That is insane.
It utilizes the tree's own chemical language to defend its fungal fruiting body from other competing bacteria and viruses in the forest. It blurs the line entirely between the host's defense and the parasite's adaptation.
A shared chemical language. I love that phrasing. Because the birch doesn't just hoard these chemicals in its bark to fight off infections. It actively pumps chemicals into the soil to communicate.
Yes.
The birch is not an isolated pillar of wood. It is a highly active node in a massive subterranean data processing network.
We have to completely shift our perspective here from the canopy down into the soil matrix, specifically looking at the mycorrhizal networks.
Fungi again?
Always fungi. The roots of the birch are colonized by ectomycorrhizal fungi. Now, unlike endomycorrhiza, which physically punch right through the cell walls of a plant's roots, ectomycorrhiza form a dense sheath, a mantle around the outside of the root tips.
Like a glove.
Exactly, and from that glove, they cast a vast microscopic net of mycelial hyphae out into the surrounding soil.
And the architecture of this fungal net is not random at all. It is mathematically precise.
Extremely precise.
When researchers, most famously Suzanne Simard in her 1997 Nature paper, mapped the physical topology of these networks in the forest floor, They found it perfectly matched a scale-free or small world network topology.
Which is a foundational concept in graph theory. A scale-free network is characterized by a power law distribution of connections.
Meaning what, visually?
Visually, you have a large number of nodes with very few connections and a very small number of hub nodes that are hyper-connected to absolutely everything else.
Oh, wow.
And what's wild is that it's the exact same mathematical architecture that governs the human brain's neural pathways, the spread of viruses in a population, and the physical routing infrastructure of the internet. It is an architecture optimized for rapid, robust information transfer.
And what exactly is being transferred over this network? Because Semart's team proved that these fungal hyphae, specifically from fungal species like Rhizopogon, physically link completely different species of trees together.
Right, they linked a Douglas fir directly to a paper birch.
And to prove the network was active, they pulsed the Douglas fir with rare, stable radioactive isotopes, carbon-13 and nitrogen-15.
Yeah, the methodology here was brilliant. They placed a sealed bag over the branches of the fir tree, injected the isotopes as a gas, and let the tree photosynthesize.
And then what?
Hours later, they took core samples from the neighboring birch tree, and they found the carbon-13 and nitrogen-15 inside the birch's tissues.
So the elements had physically moved down the trunk of the fir, across the synapse, into the fungal mantle, traveled through the mycelial network, and were pumped up into the birch.
Exactly. But the stoichiometry, the exact mathematical ratio of those transferred elements, is the smoking gun here. The carbon and nitrogen didn't just flow as raw, unbound atoms. The data showed they moved in a highly specific ratio of 2 nitrogen atoms to 7 carbon atoms, 2n.7c.
And why is that ratio so important?
Because that exact stoichiometric ratio is the chemical signature of specific amino acids, predominantly glutamine and glycine.
Okay, glutamine and glycine.
And the reason that is so profound is that in the mammalian central nervous system, in our brains, glutamine and glycine are primary neurotransmitters. They are the exact chemical signals that your brain uses to transmit data across synaptic clefts.
Wait, so under the soil, the forest is shuttling the exact same neurotransmitters through its fungal hyphae that your brain uses to think.
Yes. Alongside vital plant hormones like auxins and abscisic acid, it is quite literally a biochemical data stream. And the implications of this data stream lead us directly to the concept of forest cognition, and specifically the role of mother trees.
Mother trees. Let's talk about them.
These are the massive veteran hub trees, the oldest and largest birches in the forest. Because of their massive root systems, they are the most highly connected nodes in that scale-free network we just talked about.
And the research suggests these mother trees exhibit kin selection.
Yes.
They can actually identify their own genetic offspring growing in the understory among hundreds of other random seedlings.
Yeah.
How do they do that?
They achieve this kin recognition through highly specific root exudates. The seedlings release a chemical signature into the soil, the fungal network detects it, and the mother tree processes that signature.
Like reading a barcode.
Exactly. And once she recognizes her own kin, her behavior changes. Studies have shown mother trees will selectively pump massive amounts of surplus carbon, water, and nutrients through the network directly to her offspring, actively bypassing unrelated seedlings.
That is wild.
She can supply up to 50% of a shaded seedling's carbon requirement, keeping it alive when it can't photosynthesize enough on its own in the dark understory.
She acts as a life support system for her genetics.
Yes.
And it's not just food, it's threat intelligence too. If that mother tree is attacked by a defoliating insect, she instantly drops defensive chemical signals into the fungal network.
And a long system.
Right. Those signals travel at remarkable speeds to neighboring trees, prompting them to massively upregulate their own production of defensive enzymes and volatile organic compounds before the insects even reach them.
Now, I have to stop here and push back on the terminology a bit.
Okay.
Because when we use words like mother tree, kin selection, and communication, it's very easy to anthropomorphize the forest. We start imagining Ents from Lord of the Rings.
Talking trees.
Right. Are these trees actually exhibiting cognition and altruism, or are we just romanticizing passive chemical diffusion?
That's a very fair point.
Isn't it highly probable that this is just a mechanical source-sink gradient? The mother tree is a high-pressure source of carbon, the shaded seedling is a low-pressure sink, and the fungus is just selfishly equalizing the pressure to keep its own hosts alive. The trees might just be passive conduits.
Right. I mean, it's the most hotly debated topic in forest ecology right now. Researchers like Justine Karst have published extensive reviews urging strict scientific caution on this.
Exactly. Karst argues exactly what you just said. that we are projecting human sociology onto fluid dynamics, and that the fungus is the selfish operator, not some altruistic tree. We must be incredibly careful not to ascribe conscious intent to a plant.
Right, a birch doesn't literally love its seedling.
Exactly. But, and this is a massive, but regardless of the intent, the functional reality of the ecosystem remains the same.
Right.
Whether it is driven by conscious altruism or mechanical source sink gradients, the physical architecture of the network is functionally identical to a neural net. And the most compelling evidence that this network is doing more than just moving sugar is the phenomenon of transgenerational cascade.
Transgenerational cascade. What does that actually look like in the field?
Okay, so if you take a mature birch forest that has experienced severe environmental trauma, say a massive drought or insect plague, and you harvest the soil from that forest, complete with its intact fungal microbiome.
Okay, you got the traumatized soil.
Right. If you then plant completely naive, unstressed birch seedlings into that soil, those new seedlings will instantly exhibit altered epigenetic expression.
Instantly.
Yes, they will grow different root architectures and preemptively produce higher levels of defensive chemistry compared to seedlings grown in sterile soil.
The soil remembers the trauma.
The fungal network retains the chemical memory of the mother tree's distress, and it passes that epigenetic memory onto the next generation. That proves that learning and memory, two fundamental pillars of cognition, exist at the ecosystem level.
That is staggering. The forest is processing information, storing it, and using it to alter future behavior.
Right. So even if the birch isn't thinking with a centralized brain, the systemic outcome is a decentralized intelligence.
Which completely changes how you look at a patch of woods.
Mm-hmm.
And what I find equally fascinating is the human element in all of
I
this.
know, the anthropology is amazing.
For thousands of years, ancient humans knew absolutely nothing about DNA methylation or source sink dynamics or pentacyclic triterpenoids. They couldn't see the isotopic transfer of carbon under the ground.
Of course not.
Yet they lived in these forests. They observed the macro effects of all this microscopic biology, and they intuitively grasped the immense power of the birch. They wove it into the very foundation of human engineering and culture.
The practical applications of birch biology literally shaped early civilization in the Northern Hemisphere. Let's look at the bark again.
Let's do it.
Because of that hyper-dense betulin content we talked about, birch bark is naturally waterproof, highly resistant to microbial decay, and crucially, thermoplastic.
Thermoplastic meaning it changes state with heat.
Right. When prehistoric humans subjected birch bark to dry distillation, heating it to around 400 degrees Celsius in an anoxic environment, like burying it in a fire pit, the complex triterpenoids break down into a dense, sticky black tar.
And that tar was incredibly useful.
This birch tar was literally the world's first superglue. Neanderthals and early Homo sapiens used it to bind stone arrowheads to wooden shafts. It was the fundamental adhesive of the Paleolithic era.
And the structural physics of the bark itself made it the ultimate engineering material. First Nations peoples across the boreal forests of North America utilized the lightweight, waterproof, and flexible nature of the bark to engineer the birch bark canoe.
Such an elegant piece of technology.
It really is. The bark's cellular structure allows it to be peeled in massive continuous sheets. It allowed indigenous cultures to navigate incredibly vast, isolated, and otherwise impassable river systems, which completely shaped the trade and movement of the entire continent.
And the wood of the trunk is equally remarkable. Remember the tight, orderly paraclinal cell divisions governed by the BPVND7 gene?
The straight-grained wood.
Right. That genetic discipline creates a wood grain that is incredibly strong, dense, and dimensionally stable, especially when it's peeled into veneers and cross-laminated into birch plywood.
The strength-to-weight ratio is so exceptional that it literally built the early aerospace industry. During World War II, when aluminum was heavily rationed, Howard Hughes designed and built the H-4 Hercules aircraft, the largest flying boat ever built.
Ah, the spruce goose.
Everyone calls it the spruce goose, but that's a misnomer. The primary structural material, chosen for its unparalleled strength and flexibility, was actually laminated birch.
I didn't realize that. And you know, that exact same dimensional stability makes birch the holy grail of acoustic engineering, too.
Oh, really?
Yeah. When a speaker cone vibrates, you want the cabinet to remain perfectly rigid so it doesn't color or distort the sound. The British Broadcasting Corporation, the BBC, spent years researching cabinet resonance. When they designed their legendary LS-35A studio monitor loudspeakers, they strictly mandated the use of Baltic birch plywood.
Just for the acoustics.
Yeah, the specific density of the birch grain provided the exact acoustic dampening their engineers required.
So from prehistoric arrowheads to WWII aircraft to high-end audiophile speakers, the physical utility of the tree is just unmatched.
Unmatched.
But the human connection goes far deeper than just building materials. The biology of the tree deeply informed human spirituality and mythology across the globe.
Well, to understand the mythology, you really have to look at the birch's ecological niche. Biologically, the birch is the ultimate pioneer species.
Explain what that means in ecology.
It means it is a light-demanding, incredibly fast-growing tree that aggressively colonizes disturbed earth. After a devastating forest fire sweeps through an area, or on a much larger scale, after the massive glaciers retreated at the end of the last ice age, the birch was the very first tree to return to the devastated landscape.
It paves the way.
It does. Its tiny, wind-blown seeds can travel miles, and it can grow in highly degraded, nutrient-poor soil.
Because it is literally the biological vanguard of the forest, bringing life back to dead earth, it became the universal mythological symbol of rebirth, light, and new beginnings. Humans watched the glaciers recede, and they saw the birch follow the ice, literally bringing the forest with it.
Exactly. It's so woven into human myth. In Norse mythology, the birch is deeply sacred to Freya, the goddess of love, fertility, and renewal.
And in Celtic traditions, it is tied to Lu, the god of light. It's the central element in the springtime Beltane rituals, where a freshly cut birch tree was used as a living maypole to celebrate the return of warmth and life to the soil.
Furthermore, in the ancient Celtic Ogham alphabet, which was a language where every letter was tied to a tree, the very first letter of the alphabet, symbolizing the beginning of all things, is Beeth.
And Beeth translates directly to birch.
It does.
And then you have this incredible, slightly dark tangent into shamanism and the occult, which ties perfectly back into the fungi we were discussing earlier. We know witches in the occult are inextricably linked to the birch. The classic witch's broom, the besom, was traditionally constructed from a thick ash staff bound with birch twigs.
Right, because birch was biologically known to clean a landscape, so spiritually it was thought to purify and sweep away negative energy.
But the deeper connection is deeply psychedelic.
It involves a highly specific ectomycorrhizal partner of the birch tree, Amanita muscaria,
The fly agaric mushroom.
commonly known as the fly agaric, yes. This is the iconic fairy tale mushroom with the bright red cap and the white spots. It is a strict symbiont with the birch. Its mycelium wraps around the birch roots, exchanging nutrients. You almost never find a fly agaric growing anywhere except at the base of a birch tree.
The fly agaric is heavily psychoactive. It contains muscimol, a potent hallucinogen.
Yes.
For centuries, European witches and Siberian shamans harvested these mushrooms from the base of the birch to induce deep trance states. The legendary flying ointment used by witches and the shamanic flights to the celestial spirit world are biochemically tethered to the root system of the birch tree.
The Siberian Buryat people integrated this entire biological ecosystem into their cosmology. They literally revere the birch as the guardian of the door
The door to where?
to the other realms. They believed its roots extended down into the underworld and its canopy reached into the heavens. And by consuming the mushroom that grew from its roots, the shaman could climb the tree into the celestial realms.
It is a stunning synthesis. Ancient humans didn't have gas chromatography to map the muscimol, and they didn't have DNA sequencers to track the RDDM methylation pathways.
Obviously not.
But they were master observers of ecology. They watched the birch colonize glaciated wastelands. They saw its stark white bark reflecting moonlight in the dark, brutal winters. They observed the hallucinogenic mushrooms fruiting specifically at its base. They took precise botanical biology and translated it perfectly into a spiritual mythology of rebirth and cosmic connection.
It's why so many isolated cultures, from the Native Americans in the West to the Celts in Europe to the Siberian tribes in the East, all completely independently designated the exact same tree as the world tree.
It wasn't a coincidence.
Not at all. The ecological role of the tree is biologically fixed across the entire northern hemisphere. Human observation of that biological constant naturally led to an identical cultural reverence.
Okay, let's pull all this together because the sheer scale of what we've covered is immense. We started at the absolute microscopic level, looking at how the RDDM pathway slaps methyl groups onto the CHH sequence of the BPMYB132 gene.
Right, physically collapsing the chromatin and shutting down the flavonoid pathway to change a leaf from purple to green.
We move from the leaf into the trunk, watching how a localized drought of the hormone auxin causes the cambium to lose its spatial orientation. turning ordered paraclinal cell divisions into chaotic anticlinal divisions to create the beautiful deformity of Karelian wood.
We saw how the birch calculates its own allometric growth, staying stunted below 2.25 meters to hide its apical meristems in the thermal boundary layer of the snowpack.
We traced the mevalonate pathway as it folds squalene into lupeole and watched cytochrome P450 monoxygenases oxidize it into betulin, a chemical armor so potent we use it to trigger apoptosis in human cancer cells, and so biologically vital that parasitic fungi like chaga evolved to synthetically mimic it.
We descended into the soil matrix to map the scale-free topology of the mycorrhizal network, tracking stable isotopes of carbon and nitrogen as they flow in the exact stoichiometric ratio of neurotransmitters,
Allowing mother trees to recognize the chemical exudates of their kin and shuttle resources to them across a decentralized neural net.
And finally, we saw how thousands of years of human civilization intuitively harnessed all of this biology. They used the tree's thermoplastic triterpenoids to glue arrowheads, its dense structural grain to build aircraft and speakers, and its role as an ecological pioneer to build a global mythology of rebirth and magic.
When you look at the totality of this data, it forces a profound shift in perspective. We're used to looking at a forest as a collection of individual objects, a bunch of separate trees standing in the dirt, competing with each other for sunlight and water.
But the molecular data completely contradicts that view.
How so?
Given that a forest of birches uses chemical neurotransmitters to store epigenetic memory, share resources, recognize kin, and communicate threat intelligence across miles of soil, well, I shouldn't say ... but you get the point.
Right.
And considering that completely separate kingdoms of life, like fungi, have integrated so deeply that they synthesize the tree's exact chemical language, perhaps we need to fundamentally reconsider what a forest actually is.
Go on.
If the mathematical topology under the soil is identical to a brain, and the chemicals flowing through it are identical to neurotransmitters, perhaps the birch forest is a single, decentralized, highly intelligent superorganism.
So the trees aren't the individuals.
Exactly. Those brilliant, quiet white trunks we see peeling in the wind, they aren't standalone organisms, they are merely the antenna. The physical extrusions of a massive subterranean consciousness pushing up through the soil just to collect solar energy to feed the network below.
That is something for you to mull over the next time you step off the pavement and lean against the white bark of a paper birch. You aren't just looking at a tree. You are standing inside the muddy, brilliant chemical mind of an ancient superorganism. Catch you on the next deep dive.